Hi, and welcome to Math Listen More. Today we're going to be learning about AP Bio Chapter 3, which are the molecules of cells. So this chapter is related to Chapter 2. It's in the same unit, so please make sure to watch the Chapter 2 video before watching the Chapter 3 video. So let's dive in. Let's have a short introduction to organic compounds. Life's molecular diversity is based on the properties of carbon. A carbon atom can form 4 covalent bonds, and this is because carbon chains vary in many different ways. The simplest organic compound is CH4. Here on the bottom, you notice the structural formula, the ball and stick model, and the space filling model. So all of these show uh, 1 carbon and 4 hydrogen. So this is basically methane. All of these are methane. They are the same formula up here. So we notice that each of these have 4 hydrogens. So hydrogen, 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 and hydrogen, and they're all bonded to 1 carbon. This is the 1 carbon here, and these are all your hydrogens around it. So this is a hydrogen, this is a hydrogen, this is a hydrogen, and this is a hydrogen, and here you see a bond. And this you see the bond right here. And then in this one, here in the middle, the darker gray is your carbon, and then the other lighter grays are all hydrogens. And this is a space filling model. The 4 single bonds of carbon point to the corners of a tetrahedron. Now let's look at a few different things about carbon skeletons. Carbon skeletons vary in length. Here you notice that the length of ethane, it has 2 carbons, and the propane have 3 carbons. And thus each of the carbons have 3 hydrogen bonds in ethane, and in propane, the middle carbon has 2 hydrogen bonds, while the side carbon has 3 hydrogen bonds. So carbon skeletons vary in length. Other thing to remember is that skeletons may be branched or unbranched. So your butane is an unbranched. This is unbranched, not branched. And this is branched. So that's seen by this just being a straight line of carbon, but this one has branches here. The next thing to know is that skeletons may have double bonds, which can vary in location. So in 1-butene, here is your double bond, and in 2-butene, here is your double bond between the 2 carbons. And then skeletons may be arranged in rings. Here is where you see the ring in this cyclohexane, and in these benzene you see like an octagon-like shape of the rings. So we went over this. So carbon compounds come in hydrocarbons. So hydrocarbons are composed of only hydrogen and carbon. So here you see octane. Octane has just your carbons here in the middle, and on the outside it has hydrogens. So isomers are molecules with the same molecular formula but different structural. So in glucose and fructose, you see that they both have the same formula, which is C6H12O6, but they have different structures. For example, your double bond of carbon and oxygen in the glucose is found up here, while in fructose it is found down here. Let's quickly look at the functional groups. A functional group determines the properties of organic compounds. The functional groups are the hydroxyl group. In the hydroxyl group, you see the OH right over here. And this is oxygen plus one hydrogen. Next we have the carbonyl group. The carbonyl group is a double bond between the carbon and the oxygen. You can see it right here, and in aldehyde, and you can see it in ketone right here. The carboxyl group is a combination of the hydroxyl group plus the carbonyl group. So this does look a bit different. This is what it is, what it looks like, written spread out when you spread this out. But in the molecule, for example, of carboxylic acid, it looks like this. Next we have the amino acid group, or the amino group, and here you see amine, and your group is located right here. Next you have the phosphate group. This usually helps with your energy transfer, and helps create ATP. And this is going to be discussed in further details in chapter 5 and 6, where you learn about phosphorylation. Next is your methyl group. The methyl group consists of one carbon and three hydrogens. So cells make large polymers by joining many small monomers. The four main classes of biological molecules, so these are what biological molecules are sorted into, are carbohydrates, which are sugars, starches, and cellulose; lipids, which are waxes, fats, oil, steroids, also like hormones are proteins, muscles, hairs, enzymes, so they're a category. And then we have nucleic acids, which are DNA and RNA. Many of the molecules are gigantic macromolecules. So many of the biological molecules are macromolecules. And so on the right, we see an example of all of them. So right here, you see bread, which is a carbohydrate. Here you see butter, which is a lipid. Here you see proteins, which is meat. And here you see nucleic acids. So the building and breakdown of biological polymers. The building of a biological polymer is called dehydration synthesis. This is a condensation reaction, meaning that water is taken out. So you take water out and you put together in this process. So this is a building process. And for example, on the right, you see a glucose. And the glucose is losing water, and that creates maltose. This is an anabolism shown. So anabolism is when something is built. So here in the formula, you see that it is C6H12O6 plus C6H12O6. So you're adding two glucose molecules, and this creates a C12H22 and O11. So dehydration synthesis is basically when you join together two molecules to build a new molecule, but the product of that is water. So water is a waste of building. So breaking two molecules apart would be hydrolysis. So hydrolysis is breaking with water. You need water to break two molecules. So maltose plus water is a catalytic reaction. This is a reaction in which the maltose breaks into two molecules, and that would result in glucose. So the anabolism plus catabolism is equal to metabolism. So we will be learning about metabolism in the future in more details. So from the slides, from all the categories that we looked at in the prior slides, we're going to discuss carbohydrates in details. Carbohydrates are monomers or monosaccharides, and these contain hydroxyl groups, so that's OH, and carbonyl groups, which are carbon and oxygen with a double bond. It's important to know what carbohydrates end with. This will help you identify when you're given a name. So most carbohydrates end with ose. You'll see that when we list the types of carbohydrates. An animal's main energy source are carbohydrates. They're burned first in the body. And monosaccharides are a type of carbohydrates. These include glucose, fructose, and galactose isomers. You notice here that they all end in ose, right? Like that's seen as a pattern. You will also see that in disaccharides. So disaccharides are your sucrose, lactose, and maltose. Notice these also end with all ose. Also, please don't get these confused with enzymes. Alright, there's an enzyme called lactase, maltase. Do not get these confused. They are different things. Polysaccharides, these are created from extra sugar. These are the glycogens, which is the carbohydrate stored in an animal's liver. Starch, which are carbs stored in plants. We have cellulose. These are what the plant cell walls are made of. And we have chitin. So chitin are the exoskeletons of arthropods. One more thing to remember is that cellulose cannot be digested by animals. We have termites that eat wood, but they have a special enzyme for consuming cellulose. So, how sweet? Sweet. Various types of molecules taste sweet because they bind to the sweet receptors on the tongue. So this is a sweetness scale here. And so we're going to be looking at the compounds of natural sugars and artificial sweeteners and the relative sweetness of them to sucrose. Sucrose is like your sugar. So lactose is found in milk. Maltose is found in grains, so it's like barley, wheat, things like that. And fructose is found in fruits. So we notice here that these numbers for artificial sweeteners, the sweetness relative to sucrose is large compared to our natural sweeteners. So you'll often find these in things that need substitute for sugar. So, for example, Diet Pepsi, things like that. So aspartame is found in NutraSweet, and equals a sweetener. Saccharin is a sweet and low, so that's also a sweetener. Splenda is a drink, and neotame is very sweet. And we notice it is 8,000 times sweeter than sucrose. A very little amount can get really sweet. So this is just an interesting slide to look at. Next. So what is high fructose corn syrup? Corn sugar, which is equal to starch, and many monomers linked together is corn sugar. And so processing hydrolyzed starch broke it down into glucose monomers, which is corn syrup. We learned in the last slide that fructose tastes 4 times sweeter than to our taste buds than glucose or sucrose. So in 1970, enzymes were used to turn glucose into fructose isomers. So high fructose corn syrups are 45% corn syrup, which is glucose, and 55% fructose isomers. So why bother using high fructose corn syrup? Well, high fructose corn syrup is cheaper to make and easier to mix into a liquid than sucrose. The problems with the high fructose corn syrups were that in 1980 through the 2000s, obesity rates doubled, and as the consumption of high fructose corn syrup products tripled. There is no scientific evidence that HFCs cause obesity, but from 1980 to 2000, the U.S. per capita caloric intake increased by 23%. So now let's move into lipids. The monomer of lipid are fatty acids. So lipids reserve energy storage molecules. So what these lipids do is that they store energy molecules and they are burned after carbs are gone. The lipids are insoluble in water. They are soluble in other nonpolar solvents such as ether and benzene. This is the reason that when you have greasy hands, suppose I eat something oily, and have greasy hands, I can't get it off if I just put my hands under water and rinse them in water. I need soap to rinse them. Soap takes the lipids off because soap itself is a lipid. They're stored in concentrated amounts in the body. And they consist mainly of carbon and hydrogen atoms. And they are linked by a nonpolar covalent bond. If you need a review on nonpolar covalent bonds, you can look back into the chapter 2 video. and we have a detailed slide about nonpolar and polar covalent bonds. So there is more energy in lipids than in the same amount of carbs. So here you notice that 9 grams of lipids are equal to 9 calgrams, which is that much energy in so there are 9 cals of energy in 1 gram of lipid versus 4 cal energy in 1 gram of carbs. So some examples of lipids are triglycerides, phospholipids, steroids, waxes, oils, and fats. Fatty acids are the simplest lipid that you can find. They consist of a hydrophilic head. The hydrophilic head can actually be seen in the image up here, circled. And hydrophilic heads are consist of carboxyl groups. They also consist a hydrophobic tail of their hydrophilic head. So hydrophilic means water loving. So this head always going to go to the water, always faces the water. In the cells unit, this will be important, so pay attention. It's going to be important in the next chapter, and especially in chapter 5. It's very important, chapter 5. Alright, and then we have the hydrophobic tail. This tail here is water fearing or water repelling. Does not like water. And this is a hydrocarbon chain as seen here in the saturated fat. So saturated fats have a single carbon bonds in the hydrocarbon chain. They are solid at room temperature. And there's so some examples are butter or lard, and they have the maximum number of hydrogens. Unsaturated fats, on the other hand, have double carbon bonds in their hydrocarbon chain and are liquid at room temperature. So this would be, for example, oils. So just if you need a recap of double bonds, all of these are double bonds. So here, and then this is a single bond. So now let's look into triglycerides, phospholipids, and steroids. Triglycerides are long-term energy storage. Please pay attention to the long term. The long term actually does matter. It's important. So these triglycerides consist of 3 fatty acids and 1 glycerol. There's a picture of meat here because all of this white portion of the meat is triglycerides. So phospholipids are the major components of cell membranes. This will be further discussed in the chapter 5. So steroids, some examples of these are cholesterol, estrogen, testosterone, and progesterone. And steroids are found in 4 carbon rings. And cholesterol is the most abundant steroid in your body. It's 85% from liver, not diet. So, and waxes, they form waterproof coating. So, down here, you see cholesterol, right? And so this is a structural formula of cholesterol, and you see that cholesterol is a constituent of membranes and the source of steroid hormones. So here you see a CH chain, which is a hydrocarbon chain. And here is a picture of a triglyceride. Please pay attention to this. The triglycerides, three fatty acid, the three fatty acids plus the one glycerol. So you have your fatty acids, which look like this, the tail of the fatty acid, plus the top. So you have the three here, and then you have glycerol. Alright. So now let's learn about what trans fat is. This is also another connection, like the high fructose corn syrup. Trans fat, so an example of this is partly hydrogenated vegetable oil. In the 1890s, chemical processes added hydrogens. They added hydrogens to the double bonded carbons of unsaturated fats. Partially hydrogenated vegetable oil. This created partially hydrogenated vegetable oils. The benefits of this were that they didn't spoil as fast as others and could reheat multiple times without breaking down. So these were considered healthier than saturated fats. That's what we thought back then. So in the 1990s, trans fats were everywhere and shown to be a greater health risk than saturated fats. So you may be wondering why. So trans fats have lower amounts of good cholesterol, and they have greater or raised amounts of the bad cholesterol. So good cholesterol is known as HDL. So good cholesterol is HDL, right? Good cholesterol, HDL. And your bad cholesterol is LDL. That's bad. You don't want bad cholesterol in your system. I'm going to be changing that so that you can see the color and you can put it in your notes. And these cause a high risk for heart disease. So in 2006, there was mandatory labeling of trans fats on food labels, and since 2006, trans fats have been banned in restaurants, schools, and even in some countries. So look on the label, on food labels for trans fatty acids or PHOs. They are mostly found in canned frostings, cakes, cookies, microwave popcorn, anything that is deep fried, cookie dough, and margarine. Up here you see a picture of an unsaturated fat versus what a trans fat would look like. So this picture shows something that would be found in margarine. And this is showing something that would be found in vegetable oil. So now let's look at the proteins. As we discussed on that slide, this is one of the four groups of basic biological molecule groups. So proteins are essential to the structures and activities of life. They contain carbon, hydrogen, oxygen, and nitrogen, and they're 50% of your dry weight. So some examples of these are enzymes like amylase, sucrase, and maltase. Others are structural functions. So these are collagen and elastin. Next are contractile, which are actin and myosin. Then there are transport proteins, which are hemoglobin, which is found in your blood, protein channels, and myoglobin. Then there are storage proteins, which, for example, ferritin. And then there are hormones, such as insulin. So now let's look at the amino acid structure that is found in proteins. The monomer of protein is a amino acid. So your amino acid, each amino acid has an amino group, which is NH2, a carboxyl group, which is your uh, um, double bond of carbon oxygen, and then which is combined with the hydroxyl group. And then you have the R group. So this may be a bit confusing, but the R group distinguishes each of the different amino acids, and there are 20 different amino acids. So each amino acid has specific properties based on the R group. So linking amino acids with peptide bonds. So cells link amino acids through dehydration synthesis. We learned on the prior slide what dehydration synthesis was, and these bonds between amino acid monomers are called peptide bonds. Right here, you see that there are two amino acids, right? You have one here, and then you have one here, and they add up through a dehydration synthesis. And once they add up, they create one peptide bond here. A protein's specific shape determines its function. So a protein consists of one or more polypeptide chains. And chains are folded into a unique shape that determine the protein's functions. Protein shapes depend on 4 levels of structure, which are the primary, secondary, tertiary, and quaternary. So here you see in this animation different foldings. This is a lysozyme, and this is ferritin. So you see the different folds. Let's dive deeper into the levels of the structure. So the primary and secondary structure. The primary structure is the sequence of the amino acids. So you have the 20 different types of amino acids, and the primary structure determines the sequence of the amino acids. Your secondary structure is the coiling or folding of the chain, and it is stabilized by the hydrogen bonding. And the two shapes that are possible are the alpha helix or beta pleated sheet. So I'm just going to go through where each step is really quickly. So this is the first step that you see here. You see the amino acids coming together in a specific sequence. And then this is your secondary structure. You see the alpha helix right here. Right? This is the alpha helix. And then here you see the beta pleated sheet. So now let's look into the tertiary and quaternary structure. The tertiary structure is the overall 3D shape of the polypeptide. That's easier to remember. You can remember it by the third structure, 3D shape. The helix or pleated sheets folds due to the bond between the R groups. Here you see the tertiary structure, and you see them getting a 3D shape. And then the quaternary structure is the association of two or more polypeptide chains. So enzymes are necessary and specific to biochemical activity. We will be focusing more on enzymes in chapter 5 again. And so what enzymes do for the body, as a quick summary, is that they ease their job. They ease the job of chemical reaction. So enzymes decrease the activation energy of chemical reactions. The reactants, they work on what are called substrates. So most enzymes are named for their substrates with an ACE ending, ASE ending. And so some examples are sucrase and lactase. They are very shape specific. You can compare them to a lock and key. The reaction with the substrate with the substrate and the active site joining on the enzyme. So the active site is where the substrate and the enzyme join. So you can think of the active site as the keyhole if you're thinking of this as a lock and key. So neither do they get used up or changed. So you need to remember that they are never changed after the reaction or used up. So some enzymes need the binding of cofactors or coenzymes and vitamins after the active site. And so these help the substrate bind onto the enzyme much better. So factors that affect the enzyme activity are the substrate concentration, inhibitors, pH, and temperature. Enzymes can get denatured, which is permanent, and they lose their 3D shape. So this denaturing does happen due to your pH, temperature, substrate concentration, and inhibitors. Now let's dive into the last category of R for biological so nucleic acid. So nucleic acids, a monomer is a nucleotide. The nucleic acids help with the structure of DNA and RNA, which are the blueprints for proteins and thus control the life of a cell. One nucleotide contains a 5-carbon sugar, a phosphate, and a nitrogenous base. And at the bottom you see the structure of a nucleic acid, and you can find that right here. The sugars and phosphates form the backbone of the nucleic acid. A is deoxyribonucleic acid. DNA consists of 2 polynucleotide chains twisted around each other in a double helix. So you can see that right here in this image. And this is one of your helix chains. I'm going to do that in a darker blue so that you could see it. So this is one of my polynucleotide chains, and then this is my other polynucleotide chain, and these two combine to create a double helix. So the bases of these are adenine, thymine, cytosine, and guanine. You can see these in the middle here, and notice that you can see them right here, 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 between all the double helixes. So notice that A always goes with the T. So you notice here that the A is always going with the T, and you notice here that the C always goes with the G, and vice versa. So sugar is deoxyribose, and the bonding patterns, as we discussed, A always bonds with T. There are 2 hydrogen bonds in there, and C always bonds with G, and vice versa. And there are 3 hydrogen bonds in there. Purine bases are the A and G double ring, and pyrimidine are also bases, which are C and T, which are the single ring. And that is shown here, 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 also here. So now let's look at RNA. RNA is the nucleic acid. RNA is a single-stranded polynucleotide. There are 3 types: messenger, transfer, and ribosomal. The bases of these are adenine, uracil, cytosine, and guanine. So do you notice something here? The uracil replaces the T that we used to have, which was the thymine. So uracil replaces thymine. The in the chain here, your A would pair with the U, and C still pairs with the G. So sugar is your ribose, and purine bases are A and G, the double ring, and C and U. So in this pattern, that's what you would think, right? The A and U pairs up? No, it's changed. So I said that earlier. What happens really is your adenine is now going to pair up with your guanine, and your cytosine is now going to pair up with your uracil. So genes, which are equal to your sequences of DNA. DNA sequences spell out the amino acid sequences of proteins. There are mutation in DNA sequence, and that is a result of the wrong amino acid sequence, which results in the wrong protein shape, and then there's no functions. So let's summarize this. If there is a mutation in the DNA sequence, that will result in the wrong amino acid sequence, because your DNA tells you, your body, what amino acid sequence to put together, and your amino acid sequence tells you what protein shape you need. So what would happen is you would have no DNA sequence, like you would have the wrong DNA sequence, which would result in the wrong amino acid, and then that would result in the wrong protein shape, and then there's going to be no function. This is what happens in lactose intolerance. the mutations in the lactase gene are a result of this. So the mutations in the lactase gene, the mutation in there causes a mutation in the lactase enzyme, and then um, like lactase enzyme amino acid chain, and then there's a wrong folding in the pattern of the protein, and then this results in a defective lactase. So then your lactase enzyme isn't able to digest lactose for you, which is found in milk or dairy products, and then you're not able to consume dairy. So why is it a big deal if you don't digest the lactose in your digestive tract? Well, all the E. coli, which are the bacteria that break down your food or live in your intestines, what they will do is they will like take all of the lactose in, they'll chew on it, have a feast, and then all of their waste made from eating all this food, that feasting on all the food that you ate, will leave you with cramps, bloating, diarrhea. That's not fun. So to summarize, what happens is when you eat lactose, the E. coli in your digestive tract will have a nice feast on it, and then their waste will become your problem. You will get cramps, bloating, and diarrhea. Lactose tolerance is a selective advantage. Most people stop producing lactase in childhood, making lactose intolerance a genetic norm. In 2002, 196 lactose-intolerant adults were studied from African, Asian, and European descent. The ability to digest lactose as adults is more common in those of Northern European descent. The genetic variation to be tolerant of lactose into adulthood is a recent evolutionary change to the human genome. The ability to utilize nutrition from milk and dairy products from domesticated cattle was a survival advantage when food crops were harvested only once a year or in times of drought. In 2006, 3 additional mutations found among 43 ethnic groups in East Africa who kept dairy herds. All 4 mutations are different in sequence, but all serve to keep the lactase gene activate permanently.